Hello, everyone. Please, Giovanni, let me congratulate you and your team for these great sessions that you're organizing at Better Spain. It is an honor for me to talk after these outstanding colleagues and in front of this respectable audience. So thank you very much for the invitation. I am at the University of Oviedo. Actually, I am in the close city of Gijón, so we also have sea to enjoy. And here I'm lucky to lead an interdisciplinary team of researchers devoted to bioapplication of magnetic nanoparticles on magnetic biosensors. So if my predecessors have talked about extraordinary advances in fundamental questions of magnetism, my talk today will be very, very applied and consequently also interdisciplinary. Then probably the magnetism I'm going to mention you already know, and you may find interesting to know how we can use it to improve rapid diagnostic tests. Unfortunately, we have heard this expression a lot in the last years. COVID-19 crisis has highlighted the need for novel and improved rapid diagnostic test technologies. ADT is one way to refer to devices to detect viral proteins or antibodies generated by the patient's immune system. And the same type of rapid tests can be demanded to detect other biomolecules like cancer biomarkers or toxins, maybe toxins in foods or in fresh water. Then the World Health Organization encourages very much research on rapid diagnostic tests. So in this talk, I will explain a little bit how magnetism can contribute to an improved rapid diagnostic testing. The project relies on these three pillars. The biological target has to be determined by a microbiologist or a medical doctor. Then we develop biofunctional nanostructures, and we also develop the way to detect them. All this aiming at rapid diagnostic tests for many different applications. If you look at the applications, you understand very fast, the idea of rapid diagnostic test is not only that it is fast, which is clearly one of the points, but also that it is affordable, easy to use, easy to store, and portable, so that you can use it anywhere. Then, if you think about it, do you know any analytical device like this? I bet some of you know the classical home pregnancy test. This rapid test, as the COVID-19 rapid diagnostic tests, whether for antigen or for antibodies, is a lateral flow immunoassay. So that's the technical name, a lateral flow immunoassay. This is very easy to use. This end of the device is introduced in the biological fluid sample, and 10 minutes later, we have one red line if the test is negative, and two lines if we are pregnant. Now we need to put magnetic particles in these lines. Now let me tell you what for. But first, I will explain briefly how a rapid diagnostic test works. The lateral flow is a paper-supported system. It basically consists of a long strip of nitrocellulose membrane that acts as microfluidic platform, guiding the liquid sample by capillary action. We then, across it, print a line of a biorecognition molecule, maybe an antibody against the pregnancy hormone, for example, across the strip. This is an antibody that selectively captures the molecule of interest. And then we print a second line of a different antibody, which is not so selective. So it will capture anything biological that passes by. The target molecule is represented in my picture by a diamond, and it is contained in the fluid of the body, which can be blood, can be urine, can be saliva, that is in the tube. We will pour in this tube the nanoparticles to tag the analyte. To this end, the nanoparticles are bioconjugated to a second antibody, also specific against the target diamonds. When the test strip is introduced in the sample, one end is introduced in the liquid, then the sample flows up, and the diamonds are captured at the test line while the ambulant particles proceed until they are stopped at the control line. So one line is negative, two lines is a positive test. If in the conventional lateral flow, you see the lines in red color, this is because these tests use GO nanoparticles of 30 or 40 nanometers as tags. But we propose to substitute these plasmonic particles by magnetic particles. And why? Because with magnetic nanoparticles, we can use magnetic detection to increase the sensitivity. Look, while optical methods see particles only at the paper surface, but magnetic sensors can detect them in the whole volume across it. So that's already one advantage. Second, quantifying by optical methods is very delicate and can be challenging because there are optical interferences from the ambient light and from the paper, which can be stained or can be wet by the sample itself. And third, any detection method, including this one, can benefit from magnetic immunoseparation. So with magnetic particles as tags, we can do both immunoseparation and detection and quantification, everything at the same time. So there is a lot of room for improvement with magnetism here. Now, how must our magnetic particles be? Let me talk about 3 points: coating, colloidal suspension, and magnetic properties. The essential role of the organic capping that we must put around the particles is to provide chemical endings to attach the biomolecules by covalent bonds. That is, to bioconjugate. This is a crucial step. But additionally, an adequate particle coating can be a way to improve the surface layer quality. Well, you know very well how much spin content at the particle surface can spoil the magnetic quality and behavior. The magnetic dead layer disorder can be restored sometimes by coating the nanoparticle with an organic layer. This is not a trivial job, but it's very, very interesting thing to do. I refer you to the exciting work of Bate and Labarta and collaborators, which is mentioned here for this. But I'll tell you briefly, the idea is we're seeing here the magnetized spinal structure that lacks some oxygen atoms at the surface. When we cut the particle with, for example, oleic acid, the acid's oxygens bond to the ion in the particle, resulting in an atomic configuration which is similar to the bulk magnetite. And then the outcome is a particle with improved crystallinity and improved magnetic properties. The particle coating also contributes to colloidal stability, which is necessary because we need to mix the particles with a biological liquid. The chemical group's endings provide electrostatic repulsion to counteract the van der Waals attraction. But of course, for this, if our particles are little magnets, we also would need to counteract magnetostatic attraction. So it is much better if we use particles that do not have a permanent magnetization. Now let's go to the magnetic requirements. We need the particles to have no remnant magnetization for stability. We need them to be very susceptible so that we can magnetize them easily. And we need large saturation magnetization for detection. These requirements lead us to superparamagnetic behavior. It has it all. And also superparamagnetic sizes are small enough to interact very well with biomolecules. So everything adds up. Among the options, there are several options, of course, to detect superparamagnetic particles, but a practical RDT, the rapid diagnostic test, requires that it is something portable and affordable as a sensor, and if possible, robust and easy to use. It is why we have chosen magnetoinductive sensing, that is, Faraday electromagnetic induction based on magnetically exciting the particles and detecting their magnetic induction. Not to add complexity, not to make the device complex, we have opted for something that does not require an electromagnet or Helmholtz coils to excite the particles. That would be very complex. So we have opted for a solution of planar coils that at the same time will excite the sample and will detect the particles. And for the same reason, the inductor is fed with only a very low amplitude current. And then we continuously measure the impedance of the planar coil at radio frequency. The radio frequency is chosen because increasing frequency, of course, will give us a larger signal. We then slide the sample on the planar coil and we obtain two peaks, one corresponding to the test line and one for the control line. In a simple approach to it, we can calculate the initial susceptibility of a superprimagnetic particle using Nel model like this, the DC magnetic susceptibility, depending on the saturation magnetization and the anisotropy constant, and a term depending on the temperature and also the frequency like this. The frequency at which the susceptibility value decays, the characteristic resonance frequency, depends on the balance between the anisotropy energy and the thermal energy. So on the one hand, the particle volume needs to be small enough for this to happen at radio frequencies, small enough for the particles to be superprimagnetic. And at the same time, the susceptibility then grows proportionate to the volume. Consequently, the ideal size is the compromise that is at the threshold of superparamagnetism. Right? Then for magnetite at 20 megahertz, the optimal size is about 12, 14 nanometers. And this is the experimental corroboration of this idea. We have studied different types of nanoparticles. So among all these particles here, we have different synthesis routes and various coatings and sizes between 5 and 25. And as suspected, we see a correlation between the sensitivity of the sensor to detect the particles and their initial susceptibility, right? And accordingly, the resolution is reduced when the susceptibility is increased, right? Except for 23 nanometers, which is already very large. And then in this sample, there are already many particles which are not superprimagnetic. Then once we have the idea how the signal per particle is optimized, then we can use clustering or encapsulation in polymeric vesicles to increase the signal per molecule. And that is really something very important. Look, this is the measurement here on the right, the measurement with the inductive sensor of a lateral flow immunoassay made with 12 nanometers particles. That's the ideal size here in black at several frequencies. It is the lateral flow strip on top in this image. Now, if we cluster those same particles, then we've got this much larger response at all frequencies. The test line is, of course, more intense. And the reason for that, naturally, is that we are gathering a lot of particles per each molecule, which is immobilized in the immunoassay. So clustering is very beneficial. And for the same reason, is also excellent for immunomagnetic separation. The larger the magnetic moments, the easier this will be. Another advantage of magnetic particles, of using magnetic particles to tag, is that we can use magnetism to pre-concentrate or to separate or to isolate the cell or the molecule from the rest of the matrix, which can be complex. In this case, this is the calibration graph for a protein called pneumolysine, and let's focus on the low concentration limit to see the limit of detection. Here, the limit of detection is about 10 nanogram per milliliter of this protein. Below it, we do not have quantifying sensitivity. This is good, but we want to make it better. And if we use magnetic pre-concentration, then we can fill this gap, because all those concentrations, we bring them here to the range we can measure. So in this case, we are reducing the limit of detection by 1/10, which is even better. I'd like to show you now some results on how biodetection problems can be solved by magnetism. It's very, very exciting results. Look, imagine that for any reason your physician suspects that you might be suffering colorectal cancer. Then they will prescribe you a simple fecal blood test. This is a very sensitive technique, but very unspecific. It can be positive for many benign reasons. And if it is, your physician will strongly advise that you undergo a colonoscopy. But colonoscopies are invasive. Many people do not like them, and some people can not afford them. So what if we had a tool in the middle of the two to make a simple test, a lateral flow immunoassay, to detect colorectal cancer, even if not as conclusive as colonoscopy may be? Well, some doctors of a regional hospital asked us to do this, and they advised us to look for exosomes. The exosomes are vesicles, natural vesicles, that the cells release to the bloodstream, and they contain information characteristic of the cell of origin. I used to think it is like our garbage bags. They contain information that define us. Inside the exosomes, there is RNA and DNA, and please pay attention to the surface where we find some proteins, HE1, 9, or CD63, or proteins that appear in all exosomes no matter their origin. But then 147 is specific of colorectal cancer. Then we will use CD9 to capture the exosomes in the blood, and we will use CD63 to determine the number of exosomes of any kind in the sample, and we will tag 147 to identify colorectal cancer tumors. These are some of the lateral flow strips made with exosomes. Increasing exosome concentration to the right give us a stronger and stronger test line. These are the calibration curves with those type of magnetic lateral flow strips. In the abscissa axis, this is the number of exosomes, 10 to 7 per microliter. The curves in black here are the magnetoinductive sensor measurements, and in color, the lines obtained with a commercial reflectance reader, which is a desktop device. First thing to look at is the linear range. For the magnetic sensor, this linear range is all the measured range, while in the optical sensor, the range is much shorter in both graphs. The reason is the saturation of the immunoassay. Look, in the test line, there is a finite number of antibodies. So when all the sites are occupied, the exosomes that get there must cross without attaching. Of course, the surface layer of the paper will saturate before the volume, and it is the volume what the magnetic sensor measures. This is why the optical signal saturates before. The inductive sensor also has a better correlation factor and a smaller, so better, limit of detection in both graphs. So very exciting things for us. These are the particles we are using. There are magnetite particles coated by a double layer of oleic acid. We make it double to make the particles hydrophilic for bioconjugation. Then these strips on the left, you see the strips on the left, they were obtained in a purified sample. So from blood, it is very easy to obtain human plasma. But then to get a purified sample, we need to use ultrasiltification, ultrafiltration, and other washing procedures that unfortunately sometimes also wash the exosomes. But it is necessary. You see here with gold particles, we see two lines. So the test line is visible and the control line. And of course with magnetite too. But if we use dilate the strips in human plasma, we don't see anything with gold. But we do see both lines with oleic acid coated particles. So as you can see, If we coat the particles with oleic acid, for example, the magnetite particles provide the two lines, no sample pretreatment is needed, which is excellent news to achieve a rapid diagnostic test. Okay, the second type of application is related to beverages and foods that undergo fermentation, either in their production, like wine, beer, or chocolate, or at the beginning of the decomposition, like fish. They may contain a molecule called histamine. And large amounts of histamine may be lethal, but for some people, even small amounts can produce headache and other intoxication symptoms. I am one of those persons and I love wine. So we enthusiastically engaged in a project to detect histamine in red wine. And you can see here the lateral flow immunoassays. In this case, the assay is different. So the larger the amount of histamine, the fainter the signal. On the right, you see how wine stains the strip. So optical measure is here impossible. But with oleic acid particles, it's not only that the sample is cleaner, it's also that magnetic measurement will not be influenced by any staining. The magnetic lateral flow were calibrated again by optical and magnetic readers, and again, the magnetic reader provided the best results. So we're happy about this. It's the validation of our results with a very sophisticated technique, ultra-high performance liquid chromatography. And this was made on wines in different production stages. And you see how well the easy and fast magnetic lateral flow works in comparison with the sensitive and sophisticated technique. So to wrap up, can we use magnetism to develop a breakthrough generation of rapid diagnostic technology? I am absolutely convinced of that. In this way, and probably with other approaches. So this pushes us to make a lot of magnetic research to reduce the particle magnetic anisotropy, to increase the radio frequency susceptibility, study the interparticle interactions in vesicles, the particle interaction with the biomolecule, and the particle interaction with the sensing device, to develop a practical and sensitive technology. And with this, to face crucial challenges in health, environment, and food control. So it's exciting. With this, I'd like to give credit to our collaborators in these projects, and of course to the funding agencies, and thank you for listening so kindly. Thank you.
All right.
Sorry, I almost forget, but could you take a minute to say something about IEEE access?
Absolutely.
Oh, thank you very much. Yeah, I would like to profit the occasion because many of you are members of the IEEE Magnetics Society, but not only for those, for everyone. I want to say that recently we have launched a new section in IEEE Access Journal for the Magnetics Society. This means that now we have a gold open access journal with IEEE publishing standards to submit our papers on magnetism that will be reviewed by experts on magnetism. And you know, IEEE Access is the most competitive positions in the magnetism journal rankings. I am currently serving as the lead editor for this section, so I will be happy to receive your manuscripts and also your questions. Thank you.